Alright, welcome. I'm hoping this is a short, very entertaining video. I just wanted to briefly discuss erectile tissue and focus on the molecular mechanisms that make this happen. At the end of the day, erectile tissue is a cavernous tissue that, when filled with blood, becomes stiff. We see it in reproductive, we also see it in the nasal cavity, we see it in the ear, although we still don't understand what it's doing in the ear. But this is the take-home message. Smooth muscle relaxation, and that's going to lead to vasodilation. We can kind of see that over here in this picture. So we got a blood vessel, right? And it's surrounded by smooth muscle. And so if the smooth muscle relaxes, we can see this expand. If the smooth muscle contracts, we get vasoconstriction. And the key player in regulating vasodilation and vasoconstriction is nitric oxide. So not surprisingly, we see this in lots of systems. For example, in exercise, a key part of lifting weights, of trying to bulk up, is taking a pre-workout. Come in lots of different types. I'm not advocating anything here. But if we look at the ingredients, what we're really looking for are these amino acids, arginine and citrulline, because this is the process by which nitric oxide is generated. And so it shouldn't surprise us if we zoom in and look at the ingredients on these supplements. What do we see? Lots of arginine. The first ingredients, arginine, citrulline, with the idea being if I can provide increased levels of arginine, then I can increase nitric oxide synthesis, and it's the nitric oxide that's going to cause vasodilation. So let's take a look at the molecular mechanisms that show us how this works in erectile tissue. We're going to look at two cell types, the endothelial cell and the smooth muscle cell surrounding it. And this story actually starts off with a muscarinic receptor. Now, hopefully you're thinking, wait, I didn't think there were a lot of parasympathetics that innervated endothelial cells, that innervated blood vessels. And you're right. For most of the blood vasculature, it's only sympathetic innervation. However, we do see parasympathetic innervation in the vascular associated with erectile tissue. And the initial start is driven and by signals from the PNS, parasympathetic nervous system. This is going to be acetylcholine binding to muscarinic receptors. And we don't want to detail G-protein coupled receptor signaling. Needless to say, this leads to activation of alpha-GTP, which combine to phospholipase C on the surface. And if you recall, phospholipase C just acts as a big pair of scissors once it's been activated. And its target is a phospholipid on the inner membrane called PIP2, phosphoinositol bisphosphate. Cleavage of PIP2 by PLC yields IP3, and IP3 targets calcium channels on the surface of the smooth ER. Remember, the SER is full of calcium. So as IP3 comes and binds, the doors open, calcium spills out into the cytoplasm. Now, the target of a calcium is a calcium-binding protein called calmodulin, and it's calmodulin that activates nitric oxide synthase. And just like the name implies, nitric oxide synthase is going to take arginine, strip off nitric oxide, and the resulting product is citrulline. So the end game of parasympathetic innervation onto these endothelial cells of erectile tissue is the creation of nitric oxide. And now, like all other endothelial cells, the release of nitric oxide, which comes easily because this is small, nonpolar, non-charged, lipophilic, it can cross right through membranes, that nitric oxide is going to find its way into the smooth muscle cell, where its target is an enzyme called guanylate cyclase. As nitric oxide binds guanylate cyclase takes GTP and converts it into cyclic GMP. Cyclic GMP binds to and activates a kinase called protein kinase G, which in turn adds a phosphate to myosin light chain phosphatase. Add that phosphate group to it, activating this enzyme. Now, we haven't talked about smooth muscle and how it works, but hopefully you recognize the term myosin. Just as a brief reminder, if we look at sarcomeres, muscle has thick and thin filaments, and that thick filament is made of myosin. In contrast to skeletal muscle, smooth muscle doesn't have any tropomyosin to block this interaction, so there's always engagement with the myosin and thin filaments. However, I can regulate the level of engagement by adding phosphates through myosin light chain kinase, which stiffens the head up and allows it to bind a little bit easier, or by cutting those phosphates off with myosin light chain phosphatase. And that's what we see in this system. So with the activation of myosin light chain phosphatase, we get smooth muscle relaxation, and that leads to vasodilation, allowing increased blood flow through the cavernous chambers of the erectile tissue and stiffening the tissue. Now, we know how to turn it on. We better know how to turn it off. And that off signal comes in the form of a phosphodiesterase called PDE5. And as a phosphodiesterase, we can come cut the cyclic portion of GMP, essentially removing GMP from the system. This enzyme, phosphodiesterase PDE5, this is the target of the medications designed to treat erectile dysfunction. So we're talking about Viagra, Cialis. They're designed to come in and block the function of PDE5. With the loss of PDE5, cyclic GMP levels remain high, which means PKG remains active, which means we have increased smooth muscle relaxation and vasodilation.